Good morning, everyone. My topic is incorporation of radioisotopes in the biological tissues and cells and molecular imaging of the radioactive material. So, the atomic nuclei is composed of the protons and neutrons, and the is denoted by Z and neutrons is generally denoted by N. And the sum of protons and neutrons is called as the atomic mass, and number of protons is equal to the atomic number. So isotopes of a given element is the number are the atoms with different atomic mass but the same atomic number. That means they have a different ratio of protons to neutrons and hence they become unstable because of the unbalanced charge between the electrons and the nuclei. Hence, to gain that stability, they undergo a process called as the radioactive decay, and so they undergo the activity of radioactive decay. They are called as to be the radioactive isotopes. So there are certain methods by which the radioisotopes undergo the process of decay. The first one is called is termed as the decay by the negatron emission, in which a neutron is changed into a proton by the emission of a negatively charged beta particle, which is called as a negatron, and this reaction result in the change or the trans in or in the sorry it results in change of a neutron into a proton, but plus the release of a negatively charged beta particle. The another one is the decay of a positron. The decay results in the formation of a neutron plus a positron. Positron is a positively charged beta particle, and in this activity, the number of proton is reduced by one, and the number of proton is increased by one. This result again in the stability of the unstable isotopes. The decay by the alpha particle. Alpha particle is here released is the helium, which has an atomic mass of 4 and atomic number of 2. This kind of decay is generally seen in those elements which have a very heavy nuclei, and this result in a very considerable loosening of the atomic mass of the alpha of that heavy metal. The next type of change is the electron capture, in which the proton is changed into a neutron as the proton accepts an electron and changes into neutron. Plus, this results in the release of the X-ray radiation. The decay or by the emission of gamma rays, it is a method in which the heavy metals are broken down into lighter one by a chain of reaction, in which the alpha particle helium is sometimes produced, which breaks the element into the lighter molecules, resulting in the release of the gamma radiation. All these kinds of radioisotopes are used only when there's no other option or no other method or techniques is available, and with the utmost precautions and availability of the preventive wares. The rate of radioactive decay is defined by a rate constant called as lambda, and its mathematical equation is defined as natural log number of nt upon n naught, where nt is the number of atoms present at time t and n naught is the number of atoms present at the initial or the start of the reaction, is equal to minus lambda upon t. Time t is the time taken for the reaction to proceed. Half life of the radioactive decay is when the number of atoms present or undergoing the decay is reduced to half. It is called the time in which the number of atoms are reduced to half of the initial stage before the start or the onset of the radioactive decay is called as its half-life. The rate of decay decreases with the time. As the number of electrons are reducing, the half-life also reduces. The radioactive decay energy. The usual unit of expressing is the electron volt, and that is very small. That is 1.6 into 10 raised to the power minus 19 joules. So generally the energy is considered or it's measured in the terms of million electron volt. The alpha particles have the highest energy and they are the heaviest one, so the energy ranges from 4 million electron volts to 8 million electron volts, while the beta and the gamma have a low energy decay of about 3 million electron volt. Units of radioactivity. The SI unit of radioactivity is becquerel. This is the newer term which is generally used these days. The older unit is the Curie, based derived from its inventor Madame Curie. This is quantity of radioactive material in which number of radioactive disintegration is equal to 1 gram of the radium, and 1 curie is equal to 37 gigabecquerel. So this defines how big or how large is the radioactivity of 1 curie with respective to the 1 becquerel. Generally, while performing research activities in the laboratory, the units is always measured in the terms of becquerel, and on the large scale, where the radioactivity is measured in curie, are such as nuclear reactors, etc. These are the two different photographs which define. The first one is the types of radiation and penetration. Alpha particles being the heavier ones, so they have a low energy, low vibration, and cannot penetrate into deeper objects. Then other beta particles is a bit of higher penetration power, followed by X-ray, gamma rays, which have considerable or somehow equal penetrating power, followed by the neutrons. The energy level of the beta particles is intermediate to alpha and X-ray gamma rays. The type of another photograph or the picture here or the table shows that the symbols of the alpha, beta, and gamma particles, atomic mass, the charge present on them, the speed, and here because the alpha particles are heavier, their speed is low. Beta particles, their speed is much faster, and the gamma rays are the fastest because of the lightest one and have the maximum penetrating power. Being alpha particles being larger, so they interact with the surrounding molecules much higher as compared to the beta and the gamma rays. So the ionizing power of the alpha particles is much higher than the beta and gamma rays, and the penetrating power is lower in alpha particles, so simple paper can prevent its penetration. In the beta, aluminum foil and gamma rays can penetrate even concrete and lead. So the topic now is the incorporation of radioisotopes in the biological cells and tissues. There are certain methods of adding the radioisotopes into the cells and the tissues. The first one being the inhalation. The radioisotopes which are non-threatening or non-fatal to the human body and which have a low radioactivity are generally inhaled through the body, which are then passed to alveoli and blood, and they are reached to the specific site of their action through the bloodstream. The another one is injection. The radioisotopes are directly injected to the blood, and they reach the designated site by their circulation and of the blood, and are taken up by the cells and organs of the body. The commonly used isotope in such a case is the technetium for the radio imaging of the organs through the PET. PET is the positron emission uh, and other radioisotopes tracing elements. The micro injections are is a technique in which the radioisotopes are directly injected into the cell, and the radioisotopes used here are such as carbon, nitrogen, etc. These are used to study the cell division, cell growth, their metabolism, etc. The radioisotopes can also be incorporated into the cells or the tissues by adding or providing the radio labeled glucose, amino acid, and water, in which this particular technique is later I'll explain later. It is a in imaging for the or the for the it's imaging for the diagnostic purposes. These molecules also carry these glucose, amino acid, and water carry the radioactive sulfur, tritium, carbon, fluorine, etc., which are traced by some of the photo counters. By providing radio-labeled molecules such as sodium bicarbonate, phosphorus to the tissues and cells to take up these isotopes, these can be orally administered or by directly exposing or injecting into the body. Some of the simple isotopes can also be used orally administered. These are used to diagnose, study, and understand the metabolic action, site of disease, etc. Now the topic is the molecular imaging of the radioisotopes. The first one is the nuclear medicine imaging, in which this is a method in which the images are produced by detecting the radiation from different parts of the body after radioactive tracers is given into the patient. These radioactive images are the radioactive glucose, sodium bicarbonate, etc. For detection or the diagnosis of the cancerous or the tumor cells in the body, generally radioactive glucose is added. The uptake of the intake of the glucose by the tumor cells is far higher than at the normal body cells. So the accumulation of these radioisotopes is seen on the tumor cells, which can be detected by certain methods. Another one is the positron emission tomography, is a type of a nuclear medicine procedure in which the metabolic activities of the cells are, are measured. PET is actually a combination of nuclear medicine and biochemical assay analysis used mostly in the patients with a brain and heart condition or cancer. PET help in visualization of the biochemical changes taking in the body, in the tissue, such as for example, the change in the behavior of metabolic activities in the reproductive or the cell divisions, etc. Uh, PET is different from the nuclear medicine in the term in which PET is injected is followed by is followed after the injection of some of the radioisotopes such as technetium, and on other hand the nuclear medicine are is a method in which the radio radioactive tracers are injected into the body, and it is helped to understand the functioning of the body. Why in PET it is detect the radioisotopes within the body. That and the next topic is scintillation. Scintillation is the process of emission of light, and in this the scintillation counters are used, or counting machines are used, which detect the lightning of produced by the excitation of the radioactive material or the floors. So there are two types of scintillation counting: the solid scintillation counting, the sample is placed near to a solid floor, for example sodium iodide. The scintillation counting is a particular is in which the gamma emitting isotopes are used. Because they can penetrate the floor, the counter will be small handed. They will be counters or measured by a very small handy kind of a device which detect the emissions produced by the floor to the photomultiple tube. And on the for the large scale production, such they have a certain bench top machines which are used for the counting of the radiation or emission. In a liquid scintillation, the sample is mixed with the scintillation fluid containing a solvent or one or more fluors. So, particularly, generally, use these method is useful in quantifying weak emitters, for example weak isotopes in which the emission is comparatively low. The scintillation fluids are called the cocktails because they are the different formulation, different kind of fluors, and different chemicals are used for their preparation. For example, toluene or diisopropyl naphthalene, along with some of the fluors such as 2,5-diphenyl oxazole PPO. Another one is 1,4-bis 5-phenyl oxazole 2-yle benzene, nicknamed as pop-up, pronounced as pop-up. Or the last one, or the most commonly used, is 2,40-T butylphenyl 5,400 biphenyl 1,34 oxydiazole butyl PDP. The cocktails can be designed for the organic samples, or and this may contain some of the detergent to facilitate the counting of the aqueous solution easily and efficiently. And the certain another some of the few methods also include methods based on the exposure of photographic emulsion, the ionization radiation which act upon the photographic emulsion, or a film is produced as a latent image which is visible in the form of light. This is called as autoradiography. The general experiment or setup which is used in most of the labs is the gel electrophoresis. This is an example of the autoradiography in which the nucleic acids are separated by gel electrophoresis, and the nucleic acids are illuminated when exposed to the ultraviolet radiation. The emulsion or the film contains silver halide crystals, or as the energy from the radioactive material dissipates, the silver halide become negatively charged and is produced in a metallic silver, thus forming then it forms a layer. This layer is being monitored or observed. The photographic developers show these silver grains as a blackening of the film, and some of the chemical components called as fixers are added to the solution or that particular medium to remove that excess of the silver halide, and a permanent image is formed after the computational changes and the computational analysis of the results. Fluorography. If low energy beta emitters are used, it is possible to enhance the sensitivity of the several orders by the magnitude of by using fluorography. A fluor, as explained before, are some of the chemical compounds which get excited and later dissipate light, which is detected by the photomultipliers or the counting machines. This has been used, for example, detecting radioactive nucleic acids in gel. The fluor is infiltrated into the gel following by electrophoresis. The gel is dried and then placed in contact with a pre-flashed film. Pre-flashed film are some in which the results are very fast. So they are pre-exposed so as the film or the latent image can be seen much efficiently, I say. Intensifying screens are used when obtaining a fast result is more important than high resolution. In this, the detection of the compound or the molecule to be studied is possible when the emission is quick, and this uses the P32, that is phosphorus 32 labeled DNA, and gamma emitting isotopes. In this, the results are quick, can be analyzed, give a confirmation, but the quality and the resolution of the image is generally low, so this is not used this much these days. The pre-flashing, the response of the photographic emulsion to the radiation is not linear. So initially the results are seen in a lag phase. They are slow in nature and followed by a linear phase in which the normal results are seen. The sensitivity of the film can be increased by pre-flashing, in which milliseconds for milliseconds a flash of light is exposed to the sample before being brought into the adjacent or juxtaposition with the film, and is often used where the high sensitivity is required or results are to be quantified. So all of these molecular imaging techniques are computationally analyzed. Computers are used for this, the softwares where the graphs are plotted, and the autoradiograph results are computationally analyzed using different of the softwares. So this was all about incorporation of radioisotopes into the tissues and their molecular imaging. Thank you.